Hi, my name is Alessandro Desani. I'm one of the co-founders of the Quantum Biology DAO, and here we care about advancing the field of quantum biology, which is a proposition that life runs on quantum, and in particular, we are interested in investigating this in the context of how tiny magnetic fields affect biological processes. But we didn't really take the time to actually explore where we're at right now and what we did before this moment, sort of in preparation for all our future plans. So that's what we're going to talk about today. And this is mostly what we did in this experiment that we call the Tadpole experiment. And so let's dive into it. To give you a bit of context on the starting point when we actually designed this experiment, we were facing a lot of skepticism, both from the scientific community and from investors, on whether actually tiny magnetic field can at all do something in biology. There have been reports of weak magnetic field effects in biology for 50 years or so in many different areas, but they argued that all those experiments were not very well designed and well controlled, and so they just didn't trust them. So we took this matter into our own hands, and we decided to design a very, very convincing experiment and very controlled that could demonstrate once and for all that weak magnetic fields can indeed affect biological processes. If this is the case, if we understand how this interaction between magnetic fields and biology works, we can control biology in a predictive way by using magnetic fields, which is actually our goal for the future. And like our co-founder Jeff Enders eloquently said in a quote that you can find in our paper that covers the experiment, the reality of a given effect can be established absent a known mechanism with a sufficiently well-designed experiment. So that's what we did. We designed a good experiment to test the hypothesis that actually very tiny magnetic field can do something in biology. And let's dive a little bit into the technical details of the design of the experiment. So when designing this experiment, we always try to keep it as simple as possible to understand. And so we thought that the cleanest way of actually answering the question, did life evolve to use tiny magnetic fields like the Earth's magnetic field for functioning, was to just compare the normal condition where you have Earth's magnetic field that we always experience and give for granted, to a condition where you just remove this field and keep all the other variables like temperature, light, pressure, humidity the same. So we had two conditions, one with normal Earth's magnetic field and one where we just removed as much of the magnetic field as possible, and it ended up being roughly 50,000 times smaller than what the Earth's magnetic field is, which is already pretty tiny. So I guess now it's a good time to go over the main instrument that we used in this experiment, which is a hypomagnetic chamber, which is a very special box that we had custom made and made modifications to it. But basically this insulates from pretty much any external magnetic field and reduces the amount of electromagnetic field that is inside by several orders of magnitude. And this is done through a passive way by using special properties of materials like this mu metal and copper for shielding from magnetic fields. And also I built inside some coils that we can use for active compensation of the external field that is left inside. And so in this way you can achieve one of the lowest values of magnetic field that you can find on this planet. So we have this chamber that we use to grow our tadpoles, and we also used another box, which is an incubator, and we calibrated very extensively the conditions, and all the other conditions are very similar and comparable. So the only thing that really changes between the two is the value of the magnetic field inside. Now we need to talk about the biology part. So we do believe that these weak magnetic field effects in biology are pretty ubiquitous. They are pretty much everywhere, but we needed something to test, right? So we decided to go with Sinopus laevis tadpole embryos for several reasons. These tadpoles in particular are a very well studied model organism, so there is a lot of data and we know a lot about them, so we could compare with all the literature that has been published in the past. So that's one reason. Also, they're actually pretty easy to fertilize and grow and develop, so that even a physicist that had no previous experience in biology could do the experiment with some training. And another reason why we chose that is that they can withstand a pretty large range of environmental conditions, which overlapped pretty well with what we had in the lab. And so that was good. And the final reason is that we already had some preliminary data from collaborators that indicated that just removing Earth's magnetic field would do something on the development of these tadpole embryos. So in this case, we already had some evidence that we were going to see something. Another point is the assessment of the development of these tadpole embryos. And by keeping in mind that we want to make these as simple and clear as possible to understand, we wanted to go with something that is just clear and visible, which means morphology, or otherwise a picture. So we pretty much took a picture every single day of every single embryo, and sort of just compared what they looked like in one condition or the other. And what we saw time and time again is that the tadpoles that did not experience Earth's magnetic field actually grew faster than the ones that did experience the Earth's magnetic field. So you could ask, how sure are we that these effects are real? And the answer is actually we're pretty convinced, because we ran these experiments 7 different times with 7 different batches of tadpoles, 7 different fertilization from different parents, and each and every time we could see accelerated development in the tadpoles that did not experience Earth's magnetic field. At this point, we had analyzed over 1,000 tadpoles that always showed the same behavior. But to sort of put the nail in the coffin, we wanted to test if there was an effect that was due to maybe this hypomagnetic chamber doing something weird and unexpected because of, I don't know, the composition of the materials, or I don't even know. But so we decided to actually try to reproduce the Earth's magnetic field artificially inside of the hypomagnetic chamber, and sort of compare that to what happens in the other box with the real Earth's magnetic field. And when we did that, the effect of the difference of growth completely vanished. So the two populations were growing exactly at the same pace. And we have done this for almost 1,500 tadpoles over 3 different fertilizations. And I think this is pretty convincing evidence that even the tiny magnetic field of the Earth can actually make a difference in biology. The cool thing about this, and this is sort of how all of this relates to our quantum biology DAO, is that there are three known mechanisms in which biology can respond to magnetic fields as tiny as the Earth's magnetic field. One is through really large chunks of magnetite, so like a magnetic material that acts like a compass needle and sort of follows the orientation of the magnetic field of the Earth. But those have never been observed in Xenobus laevis embryos, and that's one of the most well-studied organisms on the planet. So not very likely. The other one is that there could be these conduction channels that sharks and rays have, but those are very complex organs, and there's no way that that is developed in an embryo that has just a few cells, maybe 30,000 cells after one day. And also that requires movement to be able to detect a static magnetic field, and over the first couple days or so, these embryos just do not move. They're just not developed enough to move. So there's no way that that is the mechanism of interaction between the magnetic field and the organism. And these only lives are quantum biology hypothesis, which means that there are some chemical reactions that depend on electron spin superpositions, and these can be influenced by tiny magnetic fields as tiny as the Earth's magnetic field. So in this case, we have a very strong evidence that a quantum effect might be at play. Okay, so in a nutshell, that's our tadpole experiment. And so the situation we're in right now is that we, I would argue, have demonstrated in a very, very solid way that even the tiny magnetic field of the Earth can do something in biology. So we know that we can use tiny magnetic fields to alter biological function. And what is left for us to do is to first demonstrate that this is actually mediated by a quantum effect that involves electron spin superpositions. And also, we want to understand how this interaction works. And once we do that, we will be able to build technologies and devices to act on anything biological, pretty much, and especially on human health. If you're interested in this, if you have questions or anything, just let me know in the comments and I will answer those either through a comment or make a video about it. If you're interested, just subscribe, follow us on all the social media, and remember that we are the Quantum Biology DAO, and we do have a governance structure that involves governance tokens that can be found on Uniswap. I'll leave the link down in the description. You can purchase the QBio token if you want to be part of the decision making of our quantum biology DAO. Otherwise, if you want to just engage with the community, you can go to quantumbiology.community, and that will take you to our Discord server where we discuss science, ideas, and everything. And as always, just remember that it isn't just science. The future of biology is quantum.